Hello. Today I will cover two topics. The first one will be glycolysis, and the second will be regulation of glycolysis. Before I go forward, I wanted to draw your attention to this slide. From now on, my lectures will be put into the self-study material in the form of a slideshow. But the Word file is also available if you wish to read the material. And I put every Word file which goes parallel to my PowerPoint presentation to the so-called basic metabolic pathways folder on Entrada. So at the end of the block, actually, you can read the Word files if you wish, if you want to review biochemistry at the same time and not going from one case to another to find the snippets of biochemistry here and there. I also sometimes put here other YouTube videos to watch. Usually those are very short, very to the point, and cover a little segment of my presentation. So for example, when I will talk today about glycolysis, there is a very simple video on glycolysis steps if you wish to review. There is also a short video on regulation of the key enzyme of glycolysis. Also regulation, how the glycolysis and gluconeogenesis regulation comes together in two parts, which is a little bit lengthier. And then one about the glucose transporters, which we started to talk about in the previous case. So let's see what I will do for glycolysis. I will introduce the basic structures and sources of carbohydrates that go to glycolysis or can enter glycolysis, the pathways of carbohydrate metabolism general, then a short overview of glycolysis, its enzymes, the mitochondrial shuttles that need to work parallel to the glycolysis, and we end up with energetics of glycolysis. The second part of this lecture will be how we regulate key enzymes. So these are monosaccharides, some disaccharides that we eat with the food, and of course we have oligo and polysaccharides that come into our body from the outside. We also have storage for polysaccharides in our own cells. So what do you need to remember, and most likely you do, that these monosaccharides, and these are just the four most frequent monosaccharides that we take from food and also produce or turn over in our body, they are either aldoses or ketoses, which means that there is a reactive aldehyde group or a reactive keto group included in monosaccharides. Thus, these molecules are reactive. This is one explanation for that you will never see a monosaccharide in this extended form or expanded form. In nature, they will actually form a ring structure because this reactive group, the aldehyde or the keto, will react one of the hydroxyl groups. So what you see here is actually a galactose molecule, and what you see here is a glucose molecule. Now, the remnant of the galactose of the ring formation will be a reactive hydroxyl group, and this also we call the reducing end of the sugar of the monosaccharide. This will react with a hydroxyl group of another sugar, so we will form disaccharides, oligosaccharides, and polysaccharides. Another important thing to remember that we have polysaccharides like glycogen stored away in our cells, but these reactive monosaccharides also will decorate lipids and proteins. So you will learn in the musculoskeletal block how glycoproteins and proteoglycans are produced. You already learned about glycoproteins and glycolipids on cell surfaces, and you also learned a little bit, and we'll learn more about how There's glycoproteins and proteoglycans, and other proteins like collagen build the extracellular matrix. So having carbohydrates in our body is very important for many, many purposes. So let me introduce what we will review in this self-study material. We will actually focus on the major metabolic pathways of carbohydrates. So what are those? If we start from glucose, we can store the glucose away in the form of glycogen, which we call glycogenesis. This is the way of producing glycogen. If we need glucose or glucose phosphate from glycogen within the cell, we will liberate that through glycogenolysis, and here it is. The most important, well, very important pathway, of course, for glucose is the so-called glycolytic pathway. So starting from glucose and ending with lactate. And when we want to form glucose in the liver, we can start from lactate or other small molecular weight components, and we can produce glucose through gluconeogenesis. And this is the pathway of gluconeogenesis, which is the reversal of glycolysis. But as you will learn soon, not every enzyme is the same in the glycolytic and gluconeogenic process. So major pathway: glycogenesis, glycogenolysis, glycolysis, and gluconeogenesis. However, there are so-called minor pathways, and one of them you already learned in the very first block, host response host depends, and that was the hexose monophosphate shunt or pentose phosphate pathway. I hope you recall, because I will not repeat it in this block, that this shunt is there to produce NADPH, which is the most important reducing equivalent in biosynthetic reactions, and also in other reactions where NADPH need to be reduced or oxidized. And also it produces ribose 5-phosphate for nucleotide synthesis, and also will produce different carbon number phosphate, sugar phosphates. This is a shunt, as you see, glucose 6-phosphate, the first intermediate of glycolysis, the starting point, and then we can put back intermediates to the glycolytic process. The other minor pathway is the galactose and fructose metabolism. I will talk a little bit about this, how galactose and fructose can participate in glycolysis, or can participate even in glucose formation. So sources of carbohydrates, as I mentioned, dietary sources are monosaccharides, disaccharides, we eat a lot of this, and polysaccharide. Starch is the main, which we get from vegetable sources, and glycogen, which we get from every mammalian cell that you can eat, if you eat food from animal sources. We have inside store of glucose, which is glycogen, and we can liberate glucose or glucose phosphate from there. Now, just 1 second, I put here, just to remember for later, that glycogen is polyglucose. Every little circle here represents glucose. Glucose can bind to each other. You remember the reactive reducing and the reactive hydroxyl will react with another sugar unit, and this linear chain is the amylose chain, while we can also build branching points. So it is alpha 1 4 glycosidic bond, this is alpha 1 6, and the branching point that we call amylopectin. We also have indigestible carbs that contains unusual linkages that we cannot hydrolyze in the digestive tract, and we eat a lot of cellulose, of course, from plant sources, which we cannot digest. Fiber is very important to eat. That also is indigestible, but very important for the health of the GI system. So this is glycolysis Everybody knows it from elementary school till now. This we consider the ancient pathway of energy generation, and it is used in every cell of ours. So this is glucose going down to lactic acid or lactate and produces two molecules of ATP. This resides in the cytosol and does not need the presence of mitochondria or oxygen. This is why all of our cells, anaerobic or aerobic, can use glycolysis for energy generation. However, energy generation is a very important but not the only role of glycolysis. Uh, we already mentioned two facts that glycolysis after the first intermediate can be diverted to glycogen synthesis, and also after the first step of glycolysis can be diverted to the hexose monophosphate shunt. Glycolysis also can supply intermediate to actually synthesize galactose if we need, or when we go further down to synthesize that 2,3-BPG, which you learned about hemoglobin in red blood cells delivering oxygen, very important to download oxygen from hemoglobin. You learned about it in the previous block, the vital gases block. If we go further, supplies intermediates for special carbohydrate synthesis. As I mentioned, a lot of derivatives of carbohydrates, modified sugars, build oligosaccharides, glycoproteins and proteoglycans. So intermediates will be taken out from this pathway for that one. Now, this can take up glycerol, which is very important because when triglycerides are used and liberated from the adipose tissue or coming into the cells, triglycerides are hydrolyzed. Fatty acid can go for energy generation, and glycerol will be taken up and utilized in the glycolytic pathway. And also it can take up fructose, galactose, other monosaccharides can join to the glycolytic process. Very importantly, it sets the stage to further process the original glucose. So in aerobic cells, lactate is not produced, but pyruvate goes to the mitochondria, and PDH, tricarboxylic acid cycle, and terminal oxidation, as you learned in the vital fluid blocks, will produce much more energy for the aerobic cell than the traditional glycolysis with its two ATPs. Just put here to remind you that we have the cell, we have a lot of compartments, and what we will focus on are cytosolic processes. So glycolysis is cytosolic. However, in aerobic cells, we will put that pyruvate into the mitochondria to produce more energy. One more thing, that when we have those specific and special modified carbohydrates, proteoglycans, oligopeptides, oligosaccharides, modified glycolipids produced, when they turn them over, they will go to the lysosomes and they will be degraded over there. So when we look at the total carbohydrate production and utilization in the cells, this is what you need to remember of bicellular localization. Now, this is a tiny little exercise. Let me switch my pointer to a pen, and let me help you to do this exercise. Now, I wouldn't mind if you would stop your video here and you would do it on your own, and then we come back, you can listen to what I am telling you. So majorly what you need to make a decision, whether in these different cells, is there mitochondria, is there good oxygenation, and whether the cells then considered aerobic or anaerobic. Now, why do I want this for you to do? Because you recall, as I mentioned before, then in anaerobic cells, it's only lactate that can be produced and 2 ATPs. And aerobic cells, aerobic glycolysis, it's called in this figure, can be run only in those particular cells that have mitochondria and oxygen. So let's say this is aerobic glycolysis, although it is not really the best way of phrasing it, and anaerobic glycolysis. Now, red blood cells, you learned about it in the previous block. You know what? Pause your video now and do the exercise, okay? All right. If you are back, red blood cells, mature red blood cells, you learned about this. In the previous block, they do not have mitochondria and they have plenty of oxygen. But you remember both of them are needed for aerobic processes. So there is no aerobic, and this will be an anaerobic cell to consider. So only 2 ATPs. White blood cells, very similar. Mitochondria, oxygen, and anaerobic. Okay, now the cells of the central nervous system, nerve cells in particular, do have a lot of mitochondria. Most of the time they get good oxygenation. If not, you pass out. So they are considered aerobic cells, which means that they will run the aerobic glycolysis. So after, by the way, the mitochondrial processes will continue. Liver, lots of mitochondria, not just for energy, but also for biosynthetic and special pathways. Better to get good oxygen, otherwise we will be in trouble. So that's an anaerobic cell with lots of energy produced by glycolysis and the continuation in the mitochondria. Cardiac muscle cells, lots of mitochondria, better to have oxygenation, otherwise you will end up with lactic acidosis and problems. So this is an aerobic cell, aerobic conditions, glucose supply, lots of energy. Red muscle cells have mitochondria. Red muscle cells sometimes have, sometimes don't have enough oxygenation, which means that sometimes it is aerobic, sometimes it is anaerobic, which means that sometimes you will produce lactic acid with the movement of your skeletal muscle and you will feel pain. White muscle cells have no mitochondria, a very small amount, and even if they get good oxygenation, and sometimes they are considered, they are considered anaerobic. So only 2 ATPs. Retina, sclera, cornea, no mitochondria for function. And although they have oxygen, sometimes they don't get enough, they are considered anaerobic. So why did I ask you again, and this is a summary of the table or the little exercise, because if a cell is functioning under anaerobic conditions, we have lactate production and we only have 2 ATPs that one glucose molecule can supply. So it is expected from anaerobic cells to take up a lot of glucose to produce the energy that they need for their function. However, when we go for aerobic cells, then they can run glycolysis, which is then continued with the mitochondrial processes, and then they will produce much more ATP. At the end of this presentation, I will calculate out this ATP number for you. Okay, very quickly about glycolysis. Glycolysis has 3 stages. One is the so-called priming stage, when we invest 2 ATP worth of energy to prime the glucose. You may recall from previous studies that if we want to enter a biomolecule into a biochemical reaction, we need to activate that molecule. So glucose is activated not just with 1, but 2 phosphorylation through investment of ATP. Then we have the so-called splitting stage, when we have the original glucose with 6 carbons and we end up with having 2 3-carbon units, 3-carbon phosphates as intermediates. And then the last stage is the so-called oxidoreduction phosphorylation stage, when we actually gain those 2 ATPs that glycolysis can supply. What do I suggest to you? There are more words for your studying, but the important information that you need to retain is set in bold. So the name of the enzyme and whether it is regulated or not. Name of the enzyme and things that you need to remember of, and whether it is regulated or not, and so on. Okay? So the very first enzyme is hexokinase. There is an isoform in the liver which we call glucokinase, and I will talk about it a tiny little bit later. You may recall just a couple of minutes ago I said hexokinase, glucose 6-phosphate, the product can be used for starting up different additional pathways, so we call it junction point in metabolism. After glucose 6-phosphate is produced, we use another ATP. So one ATP is used up, one ATP is used to further phosphorylate and activate glucose. And the enzyme which is producing fructose 1,6-biphosphate is called 6-phosphofructo-1 kinase, a mouthful. So I will refer to this from now on as PFK1. This is what you need to remember. This is the second regulated step in glycolysis. This is the committed and rate limiting step, which means from here, glycolysis intermediates cannot go anywhere else but towards pyruvate and lactate. So glycolysis will be the only pathway to go further to. This is called committed step, and rate limiting step means that this is the slowest. Committed and or rate-limiting steps in every pathway of biochemistry are heavily regulated. So this is what we will talk about in the second part of this lecture. This is the most important regulatory point of glycolysis. Second is the splitting stage. Aldolase is the enzyme, and we end up with triose phosphates, 2 of them, which can be turned into each other. Why is it important to remember this? Because 2 3-carbon phosphates will be produced, but only 1 glyceraldehyde phosphate will be used to continue glycolysis. And finally, we enter the stage oxidoreduction phosphorylation, where we will gain that 2 ATP. Okay. First enzyme is, you remember, this is one of the triose phosphates, and on the side, when we are counting on how many molecules and how many ATPs and how many NADH is produced, we need to multiply everything by 2 from this step, because per glucose molecule, we have 2 of everything. 2 3-carbon units go further in glycolysis, all right? So very first enzyme, please remember the name. You probably know this already. And high energy phosphate is produced, and also a reduced NADH is produced. We will talk about what will happen to this NADH just in a second. The next enzyme is phosphoglycerate kinase, and this is the first enzyme which will produce ATP for you. How many ATPs per glucose? 2. Because remember, we have 2 of everything here if we calculate back to glucose. Now, so we are producing high energy phosphate, and we are taking out that phosphate in the form of ATP. So this we call substrate level phosphorylation. Why I put it here? Because you already learned how to produce ATP in the terminal oxidation. this has nothing to do with mitochondria and terminal oxidation. The ATP production is done within the pathway, which we call substrate level phosphorylation. For 1 second, this is the shunt in red blood cells that produces 2,3-BPG, which is so much important in downloading oxygen from hemoglobin in the periphery. Please also note that red blood cells are not just enough that they are anaerobic, which means that they take a lot of glucose for energy, but 15 to 25% of that glucose which they take for energy will be actually used for this shunt, which means that even more glucose needs to be taken by the red blood cells. Now, next enzyme is enolase. Again, we are producing a high energy phosphate bond, which will be used in the next step, which is pyruvate kinase, the enzyme, to produce another ATP molecule, better to say 2 ATPs per glucose. So this is the second substrate level phosphorylation ATP production, and this enzyme is also regulated. So this is the third regulatory step. Hexokinase, PFK1, and pyruvate kinase. You must remember the names of these 3 enzymes. Fast forward, this will be the enzymes which cannot be reversed in gluconeogenesis. Now, you are gaining 4 ATPs. So you may ask why I said that only 2 ATPs are produced by glycolysis. The reason for that one, that 2 we invested in the previous step, so we are regaining ATP, and pyruvate kinase will be the only one which will actually produce the gain for us, the 2 ATPs. Okay, what is the end of the traditional glycolysis is lactic acid, and the enzyme is lactate dehydrogenase. You may recall that it can be a problem when aerobic cells start not to get oxygen and produce a lot of lactate. So you learned about lactic acidosis in the vital fluids block and myocardial ischemia or myocardial infarction that resulted from that. So lactate, lactic acid, and the proton that comes with it, if it accumulates in the neighborhood of the cell, it can kill the cell within the cell or outside of the cell. So it must be removed quickly and must be processed. So there are other cells like liver or skeletal muscle that under certain conditions, they can actually take back lactate and utilize it for further energy generation, reversing this enzyme, putting it back to pyruvate, and then pyruvate can go to the mitochondria and produce more energy. Now, we need to keep balance in the cells. We need to keep balance of reused cofactors. So what do we do with reused cofactors or coenzymes like NADH, which is produced in a metabolic pathway? We either need to reutilize NADH and reoxidize it to NAD in the cytosol, or we need to put this NADH energy and reoxidize NAD from NADH through the so-called mitochondrial shuttle systems, which I will talk about in a couple of minutes. So what is happening here in glycolysis, that at GAPDH level, we produced 1 molecule of NADPH, better to say 2 per glucose, and then we will reutilize it and NAD reoxidized in anaerobic glycolysis to produce lactate. So 2 NADH formed, 2 NADH reoxidized. This 2 NAD can again go back and be reused. So it actually ensures that glycolysis can run within the cell. And as I mentioned, when there is no lactate production because it is an aerobic cell and pyruvate is going to the mitochondria, then NADH will be reoxidized to NAD through the so-called shuttle systems, electron shuttles or mitochondrial shuttles. So what are these shuttles? We have two different kinds. One is called glycerol phosphate and the other is called malate aspartate shuttle. These are very smartly named after the ingredients, the molecules that participate. Both of them are between the cytosol and the mitochondria. They actually work back and forth between the two cell compartments. This is actually the inner membrane of mitochondria, which contains all these transporters, which are labeled now with white arrows, that will put through a molecule from the cytosol to the matrix of mitochondria. Right. Now, shortly, what to be known? Of course, the names. Glycerol phosphate mainly works in the muscle. Malate aspartate mainly works in the liver. They do not put NADH across the membrane, but they put the reducing power, electrons and protons. And in the cytosol, right away, the NADH is reoxidized to NAD. Okay? So not actually putting through NADH, putting through only the reducing equivalent. How? NADH is reducing a molecule. This reduced molecule is going to the mitochondria and it is reoxidized in the mitochondria, goes back to the cytosol, takes another NADH, reoxidizes NAD, and goes to the mitochondria. Okay? So the molecule which is produced by oxidation of dihydroxyacetone phosphate to glycerol phosphate is taken to the mitochondria. But please see that it is not NADH which is produced for energy generation in the terminal oxidation electron transport chain, but FADH. NADH is able to produce roughly 3 ATPs. FADH is 2. So we are losing 2 ATP equivalents, I mean 1 per molecule, if we are using this particular shuttle. The malate aspartate shuttle is much more complex. You need so-called transaminases that you will learn very soon about. And we need, of course, a dehydrogenase, which will take oxaloacetate in the cytosol, reduce it to malate using the reducing equivalents of NADH. Malate goes to the mitochondria. Malate is reoxidized and NADH is reformed. So this particular shuttle will actually preserve the NADH value, quote unquote, so 3 ATPs per NADH can be produced. Now, why do we need transaminases and why do we call it malate aspartate shuttle? Because you need, when you put the malate across the mitochondrial inner membrane and you produce oxaloacetate, this oxaloacetate cannot directly go back to the cytosol, but it needs to be turned into aspartate. Aspartate goes there, which is turned into oxaloacetate again. So this is actually a more complex shuttle. What is the energetics? As I promised, I am calculating it out for you, although you already know this very well. I just wanted to refer back what you learned about the mitochondrial processes here. So traditional anaerobic glycolysis produces 2 ATPs, and now you know the 2 enzymes where. If we go under aerobic conditions, of course, the 2 ATPs will be produced, but because we stop at pyruvate, which will go to the mitochondria, 2 NADHs will be also produced. Please note, we are calculating everything back to glucose. These 2 NADHs need shuttles to put the reducing equivalent to the mitochondria. Now, we have pyruvate. We have pyruvate in the mitochondria. It will be the substrate of PDH. And in the PDH step, if you recall, we are producing 2 NADH. So 2 pyruvate per glucose, 2 NADH. All right, then acetyl coenzyme A will go for the tricarboxylic acid cycle. And if you recall, we are producing again per glucose 2 GTPs, which are ATP equivalents, 6 NADH, and 2 FADH molecules. Now, why did I put it into parentheses? Because this NADH, total of 10, and the 2 FADH molecules need to go for the electron transport chain oxidative phosphorylation to produce 34 ATPs. If we add this together, 2 ATP equivalent is 4, 34 by oxphos, 38. Now, why certain books are listing less? Because it depends which shuttle you will utilize. You remember, with the glycerol phosphate shuttle, we are losing 2 ATPs because instead of this shuttle putting the 2 NADH across the mitochondrial membrane, in the mitochondria, in this shuttle, FADH will be produced, which means that lesser ATP. So what did you learn until now? Carbs must be taken up. Complex or simple sugars, it needs to be digested, absorbed. You already learned about it. And these monosaccharides are going to the portal circulation. So the liver is the decision-making entity, what to do with those glucose and fructose and galactose molecules over there. Glycolysis can be used in every cell of ours. And one second. And the glycolytic enzymes are all cytosolic. Anaerobic glycolysis will produce 2 ATPs, aerobic will produce 38 or 36. And NADH, which is produced, must be reoxidized by the shuttles. Now, switch to regulation of glycolysis. This is something which comes up on step 1 frequently for a lot of reasons. One, because you learned about gluconeogenesis, so these enzymes will be important. And also, glycolysis being so important in every cell, we need to define when and how glycolysis is running. Glycolysis is not running all the time, but when there is a need for that. And regulation of glycolysis will introduce you to the different regulatory factors where the whole glycolytic process can be turned on or turned off, of course, through regulating key enzymes. Okay. Regulating glycolysis through glucose availability, which means that there will be cells that can take up glucose in different ways. This is the first step. Then we have the 3 kinases. You remember, hexokinase, PFK1, pyruvate kinase in glycolysis, which all will be regulated with either small molecular weight effectors and or by hormones. Let's see glucose uptake. Glucose uptake is by so-called GLUT or glucose transporters. There are cellular differences between the GLUTs that can be on the membranes of different cells. And hexokinase, PFK1, and pyruvate kinase are the 3 kinases that will be regulated. In general, when you look at a process and you see these curved arrows, those are the steps that cannot be reversed. What does it mean? We need to have a different enzyme to go from glucose 6-phosphate to glucose or fructose 1,6-biphosphate to fructose, etc. When you see the parallel arrows, it means that these are absolutely irreversible enzymes. Okay, let's go further. Here are the GLUTs. So glucose transporter. This is facilitated transport, putting the glucose through the membrane, through the cell membrane by transformational change, not using ATP for the process. GLUT 1, 2, 3, 4, 5 are listed in the tables. In this table, there are other GLUT isoforms too. So look at this one. This is a facilitated transport. It is insulin sensitive in certain cells, and it is absolutely not insulin sensitive in other cells. Now, fast forward, insulin has no receptors on many cells of ours, but has receptors, insulin receptors, in muscle cells, adipose, and liver cells. So this is what you see in GLUT4. Among all of this, GLUT4, the only one which is insulin sensitive, and I will show you how. You remember muscle, adipose, other muscle, which is the heart muscle, and has this GLUT4 on the surface, and this is where insulin will send the signal to take up glucose. All right, GLUT1 is a general glucose transporter. These are the cells, actually almost every cell will have GLUT1 on the surface. Facilitated, not insulin sensitive, it will work down the glucose gradient. So when the glucose concentration is dropping in the cell, glucose will come through from the circulation through GLUT1. GLUT2 is bidirectional. You remember intestinal mucosal cells have GLUT2 to actually let the glucose and other monosaccharides come out of the mucosal cell and go to the portal circulation. It's important to remember that liver has GLUT2 because liver needs to actually not just take up glucose but also release it to the circulation. Kidney also and pancreas have this GLUT2. GLUT3 is more specific to the neuronal cells, kidney, and placenta. And GLUT5 is actually fructose specific. You remember the digestive tract? On the luminal surface, there was a GLUT5 which took up dietary fructose and put it inside of the mucosal cells. Muscle can utilize fructose, testes can utilize fructose, and liver definitely will take up fructose and utilize it. How is this insulin and GLUT4 in muscle and adipose tissue work? So GLUT4 is a receptor, and you see here with these little white blotches, which is not always on the surface of these cells. When insulin signals to muscle or adipose cells, one of these insulin signaling pathways will send a signal to the receptor which is stored in the cytosol in membrane vesicles to go to the cell surface, to the cell membrane, merge with the cell membrane, and expose these receptors to glucose. So a lot of glucose will come into these cells where insulin signals to the cell. Why? Because when after you have eaten, you have a very high glucose level, of course depends on how much carbs you have eaten, that needs to be cleared out. Otherwise we end up with diabetes. So high circulating glucose level needs to be eliminated, and all of the cells will work on it, but muscle and adipose have special role to take up more glucose and clear the circulating glucose level, better to say, put it back to normal. So what will happen to this glucose which comes into muscle and adipose? They will be used for energy, they will be used the extra to put it out as glycogen, and also in adipose tissue it will be used for storing away more fatty acids. Now, very important, the regulation of the three kinases. Hexokinase, PFK1, pyruvate kinase. This is glycolysis. I like this figure because time to time it shows that these enzymes are not reversible, but they will be also, at least PFK1 opposite enzyme will be also regulated. This particular figure shows you the small molecular weight regulators, which we call also allosteric regulators, and this figure shows you hormonal regulation. Hexokinase, small molecular weight regulator is glucose 6-phosphate, and both insulin and glucagon will act on glucokinase, not on hexokinase, but on glucokinase, which is the liver equivalent. So in most of the cells, this is the feedback inhibition, which will inhibit phosphorylation of glucose. So if there is enough glucose 6-phosphate for running glycolysis, no more glucose will be phosphorylated, while in glucokinase, mostly hormonal regulation will take place. This is PFK1. These are the most important small molecular weight regulators. And this is the hormonal regulation. Reminder, hormones are not signaling every cell, but they are signaling the three most important cells in carbohydrate metabolism, which is liver, muscle, and adipose. Okay, what do these little small molecular weight mediators mean? When you see AMP or ATP, which you always will see in pyruvate kinase, it sends the message for cellular energy. So when there is a lot of ATP, there is no need to run glycolysis because there is enough ATP for the cell. So both PFK1 and pyruvate kinase will be turned down, activity turned down, if intracellular energy is enough. However, when it is dropping ATP level, case AMP is produced in the cells. In some cells, it will send a positive signal to the glycolysis to run again. So ATP, AMP, cellular energy level. Alanine and citrate. So alanine you see here, citrate you see here, which are coming from other metabolic processes. They are sending the signal to stop glycolysis because there is a need for something else. So citrate is coming out of the mitochondria and it signals to the cells that fatty acids are coming to be to synthesize fatty acids in the cytosol. Fatty acids also signals availability for other energy-carrying molecules. Alanine is signaling when alanine is coming to the liver, so this is liver specific for gluconeogenesis. If gluconeogenesis is running, glycolysis needs to stop. And the proton is actually signaling for intracellular milieu, and you see it for PFK1. When pH is dropping, the proton concentration is high, then it will stop glycolysis. So for example, when too much lactate is produced in anaerobic cells, this proton will go back to glycolysis, a previous enzyme, and stops it. Now, this is the big picture, and this is mainly what I want you to know. However, I will go to a couple of details that are important to remember. First, what is the difference between hexokinase and glucokinase? Both of them are regulated. Both of them are important. Hexokinase can be found in every cell. Glucokinase is present only in the liver cell. Well, a little bit of kidney too. Hexokinase can phosphorylate other sugars, not just glucose. Glucokinase is specific to glucose only. Hexokinase has a very low Km. You remember, it's a reminder here, the Km is the constant which is typical of each of our enzyme substrate reactions, which tells us whether the enzyme is working, the enzyme is majorly saturated. The enzyme is working with half of the maximum, so the enzyme can be saturated and cannot work faster in higher and much higher concentration. It also means affinity of the substrate to the enzyme. Low affinity, high Km. High affinity, low Km. So hexokinase is saturated already or reaches this Km very quickly, 0.1 millimolar, which means that glucose comes in, turned over, saturate all the hexokinase, turned over very quickly. High affinity to glucose. So why do we need glucokinase? I already talked about high concentration of glucose circulating in the blood after we have eaten, and muscle and adipose tissue puts GLUT4 on the surface of the cell into the cell membrane to take up more glucose. And what does liver do? Liver upregulates the activity of glucokinase, which means that glucose will be phosphorylated by glucokinase, but glucokinase only saturates at higher concentration. So 7 millimolar when it is actually pretty high concentration is the Km when it is half saturated. This mechanism also takes a lot of glucose inside, and it also gives an opportunity for the liver to channel this glucose 6-phosphate into different liver-specific metabolic pathways. So this is the point when a lot of glucose is coming into the cell to use this glucose to store away in the form of glycogen, also convert it to fatty acids, and to produce modified sugar residues. This hexokinase regulation is actually a little bit more complex. I wanted to say funny, because we need the enzyme when the glucose is coming into the cell in more concentration. Do you observe GLUT2 here? But this enzyme is not phosphorylated or dephosphorylated. This is actually stored away in the nucleus. And when glucose is coming in larger concentration to the liver, that gives a signal to the enzyme glucokinase to go to the cytosol and start phosphorylating glucose. Now, glucose came in, lots of them phosphorylated. It needs to go down in the glycolytic pathway, or beforehand goes to glycogen and hexose monophosphate shunt, if you remember. But when fructose 6-phosphate is accumulating, you remember the next step is PFK1, which is the rate-limiting enzyme. So fructose 6-phosphate will be accumulating. It sends the message for glucokinase to go back to the nucleus and not phosphorylate more glucose. Pretty fine, is it not? Really nice method to regulate how much glucose is going to be phosphorylated. Now, I put here something, fructose, and why those high fructose syrups and sodas and whatever is actually bad for your health. So fructose is coming into the liver cell, no regulation there. There is a specific enzyme which we call fructokinase, which will phosphorylate fructose to fructose 1-phosphate, no regulation there. This fructose 1-phosphate can go down in a separate pathway, but before it goes, fructose 1-phosphate also will send a signal to glucokinase to come and phosphorylate glucose, which means that when fructose is pouring into the cell, glucose will be phosphorylated and more glucose will come into the cell. What will happen with the glucose? Let's say we don't need more glycogen. We don't need more hexose monophosphate shunt products. We don't need more modified sugar products. So it will go down the glycolysis and it will result in producing fatty acids. We are gaining weight when we eat more fructose than necessary. So it can lead to hypertriglyceridemia. Bad. I put it here. Do not eat drink products with high fructose corn syrup. Look at the label. All right. Very quickly, because I already went through of this, this is very important to remember, okay? Second regulatory enzyme, committed step, rate limiting step, and all of the effectors. Allosteric, ATP, citrate, proton are negative, and allosteric activators, AMP, fructose 2,6-biphosphate. This is important. Hormonal regulation of this particular enzyme will actually go through producing fructose 2,6-biphosphate, and this is insulin signal, or not producing fructose 2,6-biphosphate, which is glucagon signal, glucagon or epinephrine signal. Let's see the details. Before we go there, please remember PFK1 is a glycolytic enzyme which produces fructose 1,6-biphosphate. PFK2 is a regulatory enzyme that produces fructose 6-phosphate, the allosteric activator of PFK1, without which glycolysis would not run, because PFK1 is the most important regulatory enzyme. So how do we produce fructose 2,6-biphosphate, which is such an important regulatory, positive regulatory factor of KFK1? This is produced in an multi-activity enzyme. So which means that the same enzyme has two activators. One is a kinase and one is a phosphatase. Kinase and phosphatase units are residing on one enzyme, which we call PFK2. PFK2 kinase activity, PFK2 phosphatase activity will determine whether we produce fructose 2,6-biphosphate or we eliminate it. This will be the insulin signal, and this will be the glucagon signal. So, what is the action of hormones on PFK2? In the liver, glucagon, which upregulates kinases, will phosphorylate the kinase unit. The kinase unit is inactive, thus the phosphatase unit is active. So in glucagon signal, we do not produce fructose 2,6-biphosphate. In insulin signal, we take off this phosphate group, we dephosphorylate the kinase, which then turns into active. The phosphatase turns into inactive. Thus, we produce fructose 2,6-biphosphate. There is a little difference here in the heart muscle. In the heart muscle, this particular enzyme, PFK2, has the phosphorylation site in the phosphatase unit. So when we are under stress and epinephrine is signaling to the heart, it will upregulate phosphorylation, but this phosphorylation will inactivate the phosphatase unit, activates the kinase unit. So fructose 2,6-biphosphate will be produced. So glycolysis will be upregulated, energy will be produced for the stressed heart. Glucagon epinephrine upregulates kinases that phosphorylate proteins. Insulin upregulates phosphatases that dephosphorylate proteins. Remember, which I just said before. Now, this is just a tiny little bit more complicated figure, and it says that we have a glucagon that signals to the cell, upregulates a kinase. The kinase is phosphorylating the PFK2 kinase subunits, and fructose 2,6 biphosphate will not be produced. It means that glycolysis will be stopped, and just fast forward, gluconeogenesis will be allowed. Insulin signals to the receptor. Insulin upregulates phosphatases, which will take off the phosphate from the kinase subunit of the bifunctional PFK2, which means that fructose 2,6 biphosphate will be produced. Glycolysis is allowed to run. Okay, summary: glucagon kinases phosphorylate PFK2, inhibit the kinase subunit, no production of fructose 2,6 biphosphate, no stimulation of PFK1, glycolysis stops. Please remember, this is liver. Insulin phosphatases dephosphorylate PFK2, activates the kinase subunit, produces fructose 2,6 biphosphate, which stimulates PFK1, glycolysis runs. Please spend a couple of minutes of remembering this mechanism. And finally, the third kinase, pyruvate kinase, how is it regulated? Inhibited by ATP, inhibited by alanine, which is the signal for the liver that gluconeogenesis needs to be done, and activated by fructose 1,6 biphosphate, which is produced in glycolysis earlier by PFK1, and it sends the signals to this PK enzyme a little bit later to be ready because molecules are coming down in the glycolytic pathway. Hormonal regulation of PK is also important. It is now a different way. And this is actually more of a typical way of regulating enzyme activity by phosphorylation and dephosphorylation of the enzyme. So the PK is active in the dephosphorylated form. Glucagon upregulates kinases will phosphorylate the pyruvate kinase and it turns it inactive. Insulin with the phosphatases upregulated dephosphorylate the enzyme and activates PK. So insulin is the signal to make this enzyme also active by dephosphorylating it. Please also remember, insulin also works in a gene expression level and upregulates the gene expression of this enzyme, which means that more enzyme will be available to run glycolysis. While glucagon downregulates this gene expression, less enzymes will be available in the cytosol. This is just for your information. PK deficiency results in hemolytic anemia. Why? Because PK is at the very end. You remember that in red blood cells, this is only the anaerobic process, only the 2-ATP that produces energy. 2-ATP are invested to recovering this investment, and only PK, which will produce those extra ATPs which can be used by the cell. So if PK is not working at a normal level, there will be not enough energy for the red blood cells. 5 to 25% of the residual activity of PK remains, then pyruvate and lactate levels are going up and red blood cells will lyse. If you have more than 25% of PK in the cells, that's a lesser problem. There is no problem in reticulocytes. Why? Because reticulocytes have mitochondria, which means that lactate will not be produced, but pyruvate will go to the mitochondria and much more ATP will be produced. So even in patients with PK deficiency, the disease will not be manifested itself in the reticulocyte level just only when the red blood cells mature. This is a very good summary slide which you can use to remember what we talked about today. And summary for the regulation is that we talked about the different glucose transporters as one of the way of regulating glucose entry to the cells. And we talked about the three kinases, how they are regulated by small molecular weight effectors or by hormonal regulation. Please remember, hexokinase, glucokinase are isoenzymes. The most important regulatory enzyme is PFK1, and pyruvate kinase is the third one. Thank you.